Дамы и господа, давайте начнем.
Ladies and gentlemen, let's start. I will invite the experts to take their seats up here.
Устройство для...
Make sure you have the simultaneous translation kits.
Next generation of communications that brings together neurointerfaces, collective collaboration, and semantic translation, art, and all other forms of body and mind communications. We have experts here. We have Karen Casey. She's a director and visual artist from Australia, famous for her Global Mind project. That is a project. This is a visual art project that brings brains into collective art communications. This is brain art and music, as far as I understand. It's very interesting, and it helps us to bring into this theme a new aspect, because all other experts are more deeper into science part of our talk. So we have also Pavel Luksha, expert from Moscow Business School Skolkovo, a leader of Global Education Futures project, and he will be experting this topic as well, because he is co-founder of NeuroWeb group. We have also Stephen Dunne here. Stephen Dunne is a leader of Starlab laboratory. This is a better, I think, in many ways, equivalent to MIT labs in United States. This is a laboratory that leads whole bunch, I would say, of different projects connected to collective neurocommunications, to robotics, to space, and to many different areas. I think Stephen Dunne has the most wide area of implemented projects now, and very interesting business model, because the Starlab is deeply implemented into European programs of working with high technologies. So we have Mikhail Lebedev here, and he is from Duke University. You might know him from this very famous experiment when monkey takes a banana with a robotic hand controlled by her EEG. So he is a author of this experiment, and he is one of the world leading experts in this area. And finally, we have Dr. Randal Koene here, who is a COO and a founder of the Carbon Copies, and also a head of several startups and businesses that are working with whole brain emulation issues, and with actually creating copies of brains in non-biological substrates. So we will now start from Dr. Koene presentation, and we will be asking questions and try to make it very interesting and useful conversation now. So I am now heading the moderator role to Dr. Koene. Thank you.
Thank you. And I think this microphone works, so I can work with that one. Is that a clicker for the slides? Thank you. Okay. Yeah, so it looks like the order in which we're going to do the speakers takes us from the most futuristic backwards towards the most near-term, most practical and applicable, with mine being probably the most futuristic of the talks here. I think if we're talking about the concept of neuroweb or neuronet, a very important aspect about that is to connect minds, to connect a mind or the minds of multiple persons in one way or another. Of course, if you're going to do that, then the most primary and important need is that you be able to make these kinds of connections, to have better access, to make for more creativity, insight, and well-being. And so we're going to start looking at this problem from a number of different angles with the different speakers that Timur has already introduced to you. And let me therefore start by telling you a bit about my contribution to this, so my particular angle of looking at the problem. And I don't know where I have to point to get the clicker to understand that it should move forward. Oh wait, I'm doing it the wrong way. There we go. That's better. All right. So I'm going to start off with some useful terminology to explain where I'm coming from. Actually, if you want to read a lot about what I do and who I am and what this is all about, this whole idea of what I call substrate-independent minds, whole brain emulation, and all that stuff, there was a recent article in Popular Science that explains it very well. It's a well-done article. Or you can go to that website, koene.carboncopies.org. I can tell you a little bit about my motivations. Basically, it started from a point of creativity, actually. I used to like to do a lot of creative things, writing and making music, many things like that, and kept bumping into various limitations, either time or something that I felt wasn't easily done by one person or people couldn't do at all. And so thinking about that, it occurred to me that all of this comes back to what's inside the mind. What are we capable of doing? How long can we even follow through a project that we have? How many things can we hold in our mind at the same time? What sort of senses are we able to perceive? And when you think about that, then, well, what do you need if you want to improve that? If you want to improve any of these things, you need to be able to get access in some way or another. You either need to read something from the mind to understand more about what's going on inside of it, or you need to be able to write something to the mind, stimulate the mind in some way, bring something into it. So getting access is hard because biology is not intended for that sort of access. Our neurons are not built to be readable from the outside and writable. Of course, there are methods that you're going to hear a lot about from the next speakers that try to do that with the biological brain. But looking forward, what occurred to me and to many others as well is that ultimately, if you take this all the way through, if you wanted to have complete access, the best way to do that is if you have the mind in some substrate that is perfectly accessible, where you can read everything and write everything. So this is kind of where I'm coming from. I call that substrate-independent minds. So the goal is, can you take what the mind is doing, the way it's running on the brain, and move it to some other substrate where the same functions are running? It's not quite like artificial intelligence, because the intent there is really to take what is in this brain, in the mind, move it somewhere else. Another way of looking at that is in terms of what they call neuroprosthetics. Neuroprosthesis is the attempt to build a replacement part for a small piece of the brain. Now, if you do that for all of the pieces of the brain, that's when you get to whole brain emulation. That's the next term here. Emulation meaning, let's say, for example, when you have a program that's written to run on a PC, and you can run it on a Macintosh because the Macintosh is able to emulate the environment of the PC. Okay, so these are the terms that I wanted to mention. And when you move something from biology to this other substrate, the term that is used for that, a popular term, is often mind uploading. There's also a project here in Russia, the 2045 initiative, that has a different term for it. They call this the Avatar project, and Avatar C is their term for this uploading process that happens there. Now, for humanity as a whole, I think the interesting thing about this is that what it does is it is a way of achieving what I would call self-directed evolution, the ability to move out of a specific niche that we were evolved into, selected for certain challenges at a certain time in a certain environment that isn't always going to be true. There's a reason why people don't just travel into space without a spacesuit. We have to take the environment with us wherever we go. There's a reason why we can't process data in the same way that Google can, because our minds weren't built to deal with that much data that fast. So there are simply some limitations that are imposed by the way we were evolved from nature. Okay. Now, if you want to get started with this, even to just think about how to do this, one thing you have to realize at first is that we can look at the brain as a type of machine that is running something, that is doing something, producing something, from which we have emergent cognitive functions. And we can identify operations in there, and we can interact with those in some way. So in the next slides, I'm going to show how you might do that, how you could actually treat this as a machine that you can analyze, and where you can take the functions of what it's doing and move it somewhere else. And we're in luck, because this really is the era of neuroscience and the era of the brain, as you'll see from some of the projects that are going on. Okay. Now, it's important to realize just how primary the brain is to everything that is human experience. Really, what it comes down to is, for us, nothing exists except what is being processed in the mind. I can't really touch this table. All that's happening is that atoms are getting fairly close to each other. They never actually connect. A force is exerted upon some sensors that are then producing an electrical signal that goes up into my brain. And when it's processed there, then I become aware of something, and I interpret that as I'm touching something. And I put that into my worldview of what I'm seeing here. I'm touching a table. But if there's something that I'm not aware of, something that's not being processed in my mind, it may as well not exist to me. So this is just how central the brain is to our human existence. And what we have now is an opportunity to do something to overcome various kinds of limitations and incapacities, or to deal with diseases, etc. there. So what kind of limitations do I mean? The brain is a very wonderful thing, but of course we have a number of limitations, some of which I already mentioned just before, but even simpler things, such as our working memory, is very limited. We only have a span of, some people say 3 to 4 items, some people say 5 to 7. It depends which type of short-term memory you're talking about, or working memory you're talking about. We have a long-term memory that forgets. It does not remember perfectly. It certainly doesn't remember, like, taking a picture of something. There are many other limitations like this. So what can you do to achieve something like a neuroprosthesis? Well, neuroprosthesis means being able to replace parts of the brain by looking at the function of what's going on inside of it. And can that be done? Well, we have some evidence that it can really be done. I like to give this example because it at least gets over the first hurdle of thinking, wait, this is so different from a computer or something else. We can't just replace parts in it. Yes, we can. There's this example of the hippocampal prosthetic. It's a cognitive neuroprosthetic developed by the Berger Lab at USC, that's University of Southern California. It's not a very fantastic device. It only replaces the function of the hippocampus within a specific experimental setup where either rats or primates have to carry out a specific task that requires memory, and the hippocampus is very closely involved with the ability to generate new episodic memory. That's memory of sequences, such as your ability to follow what's going on in this talk. And what he was able to do is he was able to put electrodes on one side. Oh, is this it's not working? Oh, there it is. Yeah. Actually, I can't really point at the screen. It's not showing up. But if you see on the bottom picture there, you see these 2 blue plates. Those are electrode arrays that you have around an area of the hippocampus where there could be damage, for example. And if you're recording on one side of that, you've got input, and on the other side you have output, then you can use a process called system identification to monitor what are these inputs and outputs when the hippocampus is working properly, and what could be the transfer function that explains how this output is generated from the input. And by input and output, I only mean the spike timing, the timing of the activity of the neurons. When does a neuron fire? Just looking at that was sufficient for Berger to come up with a model that incorporated such a transfer function and that could be put into a chip that was able to replace the performance, make the rats be able to perform the memory task in that experiment, or the monkeys in their experiment, and he's now got a grant to go and carry out the same sort of research in humans in 3 to 4 years. So this is telling you something. This is telling you that, in principle, it is possible to treat the brain in that fashion. It's not necessarily always going to be as easy as what he did there, but this is just a proof of principle. Okay, I already explained what system identification is, so we can jump over that. But if you're going to do this for the entire brain, you have the problem that it's a very large system. You can't just look at the input to the entire brain and look at the output of what we're doing with our motor system and say, I'm going to watch it for a while and I'll come up with a transfer function for all of that. That's simply not going to work. But what you can do when you have a big problem like that is you can break it down into very many small problems, into a lot of subsystems, each of which is hopefully not much more complicated than what Berger was looking at there, to then look at it and say, I'm going to characterize what's going on in that subsystem. So what do you need to do for that? You need to find what are all these subsystems and see how they're connected with one another. You could do that at many different levels, but one level, is to look at how are all the different neurons connected. And that's called connectomics. It's looking at the connectome, the connections within the brain. So what we've got here now is we've got a roadmap, basically. How could you get to whole brain emulation? What you need is you need a way of looking at the structure, how various parts in the brain are connected to one another, maybe how every neuron is connected, maybe even smaller details, such as what are all the little dendritic structures in there. You also want to be able to characterize these individual systems. So you need to do system identification for them. And right now, the only way to get a fairly good idea of what is the activity in each part of those systems is to actually record that activity. So that means you need to do high-resolution, large-scale neural recording. So you need great methods of recording from the brain in vivo. Then you also need to be able to take all these recordings and put them into a model that actually works. Just having a bunch of data is not enough. You need to know what it means in terms of parameters so that you can put it into a function and run that. That's another important part of that roadmap, exploring those functions. And then finally, of course, you need to validate, because you're starting with some initial model. For example, you assume that spike timing is enough, and you assume that you can record from every neuron, and that's going to be enough, or something like that. Maybe something a little more abstract, maybe more detailed. But you're going to run into things that work and things that don't work, and then you need to dive deeper in some areas, not as deep in others. Iteration, validation is very important. Okay, so a quick view at some of the technologies that are being developed or already have been developed in these different areas. So for connectomics, just to give you some idea of what some of them are, one approach is to use DNA barcoding to try to find out what is connected to what. This is a method that is being pioneered by the Tony Zador lab. And what it is basically, the idea is take a piece of synthetic DNA that has a certain code, insert that into a neuron. Now do the same thing with different pieces of DNA for all the other neurons. And then use something that can carry that DNA across a synapse into the next cell. For example, a rabies virus or some other virus that is more suitable for this, that will only cross one synaptic barrier. Now, they have done a lot of research into finding the right vectors to transport, and I think that's getting somewhere now. So what you have then is it's like giving postcards to all of your neighbors. If you look inside a single cell and you take out all these strips of DNA and you sequence them, you know which other neurons they came from. So this is giving you an idea of the connectome. Although all you know is which neurons are connected with which other neurons, you don't know how strongly, whether it's inhibitory, excitatory, all sorts of other details like that. There's another kind of looking at connectomics. It's a slightly less high resolution, for example, using optical volume microscopy. The picture you see here is from the Clarity protocol. It's a protocol that removes the lipids in the brain so that it becomes transparent. That's useful for some types of study, but not really high resolution enough for much of what I was just talking about. So I'll move straight on to using electron microscopy to find the connectome. This is something that is being done in very small pieces of brain right now, and it would be nice to be able to expand that to entire mouse brains or larger brains than that. But right now we're talking about little volumes, maybe a micron by a micron by a micron, or a few more microns, maybe 10, 100 microns, something in size. And at that, you can, with an electron microscope, you can do scans going through this slice by slice at a resolution of up to 5 nanometers for the type of scanning they're doing here, which is enough so that you can identify individual synapses and even the vesicles that carry neurotransmitters inside those synapses that give you an idea, for example, of how many vesicles are near the surface and therefore how strong is that synapse. From that, you can reconstruct things like this network that you're seeing on this picture here. We're showing you the neurons and showing you the connections between them. Now, let's head into the functional stuff. That is an area that hasn't developed quite as far. The connectomics has had some really nice results. I didn't talk about the beautiful proof of principle papers that came out in Nature in 2012. I won't get into that right now, but Connectomics has developed for about 5 to 7 years now, since it became a really serious area of development. This, looking into high-resolution functional recording, is just now really taking off, especially with the so-called Brain Initiative that was started with the idea of developing exactly that. One way of doing that is to just continue down the path that we're already on, using arrays of electrodes to record from the brain, and to use many more electrodes on those arrays, and to, for example, include in that optical guides, making optoelectrodes so you can do optogenetics, targeting specific neurons, turning those on or off, and then using recording from those neurons. The arrays are getting quite large. We've had arrays of about 10,000 electrodes that have been developed at the the Boyden lab now at MIT, and they're planning to go up to 1 million, which is a lot when you think about the fact that a mouse brain contains only about 20 million neurons. Now, another way is using fluorescent microscopy, which I won't get into right now, even though it's really fascinating for using that on small animals. More interesting to talk about for the moment is the so-called molecular ticker tape. The idea there, it's kind of like you saw the Tony Zador approach for getting the connectome. Again, we have a method using DNA barcodes. In this case, small strips of synthetic DNA inserted into a cell, amplified so that you get a longer strip that keeps repeating the same code. But you also have a channel that makes sure that when the voltage changes in the cell, that the error rate in the amplification of that DNA changes in accordance or correlated with the activity. So if you were to pull out that DNA and you look at it and you see where the errors are, you get an idea of what is the activity that was going on. It's currently being tested in George Church's lab at Harvard. There are some issues with getting a good sample rate, with getting reliability, how many of those tapes do you need per cell, etc. But it's a good technique in the sense that biological techniques are easily applied to many cells at once. Now, another interesting method is to try to do what we're doing with electrodes, but make it wireless. Have really tiny electrodes that can record from inside the brain, and that are perhaps so small that you could have as many as you need to be able to record from all the neurons there. This is being studied by several labs. There's one at MIT that is doing this with infrared. There's another one at UC Berkeley that is using ultrasound as the method to communicate with these wireless electrodes. That method there is called neural dust. They have a prototype right now that's at 126 micron in size, and they're hoping to shrink this down to 20 micron. The advantage of ultrasound is that it doesn't heat up the brain very much when you're using ultrasound because it's not absorbed much. Yeah, this is a good bit of progress. And I'll tell you a little bit about how this all comes together. So if you're trying to do something like whole brain emulation, a really complicated project that has a roadmap that spans so many different areas, one of the problems is that you deal with many labs that have their own incentives that are being motivated by the grants that are available. How do you know that things are going to end up going in the direction you're trying to do for a long-term big project like that? Well, I like to look at it as lots of little vectors that are pointing in many different directions, and if you add them together, you get trends, but the trends don't necessarily follow your goals. So what can you do to push these little vectors in the directions that you want them to go? I mean, aside from just offering grant money, there are other things that you can do, for example. So one of the things that I do through my nonprofit organization, Carbon Copies, is that I stay in touch with all these different laboratories, and I talk to them about these roadmap issues. I talk to them about what a person over here is doing and a person over there, trying to get problem people together with solution people, trying to find funding when funding is difficult to find, making sure that the roadmap is updated so that it's always clear where are the new gaps that are appearing right now, where are things not being looked into at all. So both looking at it from bottom up, what are people doing, and top down, what is necessary. And that does lead to some interesting results. I've made a very complex looking slide here that just shows you in circles, kind of the circle, the network, or some of the people in the network that has come about that way. You can see in those 4 squares, structure, function, emulation, and application validation, again, the pieces of that roadmap that I was showing you, the important pillar areas that you needed to look into. In that, you can see a core, the smaller circle, that I call core partners, where the people involved are actually interested in the same problem. They would love to see neuroprosthesis accomplished, or neuroprosthesis for the whole brain, or lots of parts of the brain, or whole brain emulation. And then outside of that, there are people who have similar goals technologically. They need it for some other purpose, but they're not necessarily interested in exactly the same goal. And over the last few years, I've seen these circles expand so that core partners, more of the people outside of that circle become core partners after a while. And some of the names in here you might recognize, but I don't have time to get into that in detail. Just to give you an example of how that works, for instance, a few years ago in Silicon Valley, I was working in a company that was interested in DNA sequencing. And so I invited Ed Boyden from MIT over to come and give a talk there. And we started talking about how could we use DNA sequencing as a way to address the problem of recording from the brain. This is kind of where he started thinking originally about this molecular ticker tape idea. He went and talked to George Church, and they had Conrad Cording from Northwestern involved, and this became an actual project. Plus, around that, they built a group called the Physics of Brain Activity Mapping Group. Their specific and explicit goal is to come up with technologies that can record from every single neuron in a mouse brain at 1 kilohertz sample resolution. And for that, they write white papers and published papers on the different technologies. What kind of hurdles did they run into? How can you combine different ones as hybrid solutions to get around those hurdles? So that really took off. In addition to that, this other guy down there, Jose Carmena from UC Berkeley, came to me one day and said, Randal, we've been talking about this whole brain emulation thing for a while. At first, I didn't think it was such a great idea. It seemed way too far out there. I've read some books, and now I think it's really interesting. And then I told him about what Ed was doing, and he said, I knew this guy, and we always talk about, Alzheimer's disease grants and stuff, but I never talked to him about this. And he got in touch with that group, and it turns out that their work on neural dust fit perfectly with what they were doing at the Physics of Brain Activity Mapping Group. Plus, that group, the Physics of Brain Activity Mapping Group, was then one of the main groups that put together a proposal that was then sent to the White House and eventually became the Brain Initiative. So, little pushes here and there, little bits of work, but ultimately it has an effect. And I'm very happy to see what kind of results it's had. For example, this thing, the neural dust prototype, which I already told you something about, and that is now in animal trials. So, yeah, there are things you can do to make a project like that move along. Now, another issue that was important was to notice that the way that the funding works, the way that the incentives work in academia is not usually the method that you very rapidly get improvements in the same technology to the point where you have something that is reliable, that is usable beyond the research stage. A better way to get into that is if you have something commercializable, something that has an interest where you get rapid feedback. And that's not possible with something as far out as whole brain emulation. So the clean room version there doesn't work that great. But the better way to approach it is to use a piecewise approach. If you look into brain-computer interfaces and you notice that the really cool applications that can come out of that are the sort that require also high-resolution, in vivo recording, you see that they need the same types of new interfaces. And therefore, that's a really good way to proceed. I'm almost at the end of my talk, so don't worry. I just wanted to, I'm going to jump over this. There's an application timeline that comes from looking at all these new technologies that they're developing out of there. I just wanted to mention that in addition to doing the nonprofit stuff, what I also did is because the BCI angle seemed like a really good thing to push, I started a startup, NeuraLink, where the aim is not to build another interface, because there are already different groups doing that, and you don't want to bet on a single horse, but to make the improvement easier by making protocols reusable. So to get away from having to redesign and reinvent the wheel every time when you have a new device, setting up new infrastructure around it, new software around it, but to make a platform that is more like an operating system where you can plug in different devices, different applications, and then in the future, when these devices become ever better, you can have some wonderful possibilities for brain hacking, as I wrote down there. I'm not going to get into detail because I don't have time. But yeah, I'd be looking for Russian partners who are interested to also share their protocols and see if we can integrate that with that platform. And let me see if I can finish quickly. OK, the last thing I wanted to mention is that, as you saw, what this is all about is cross-disciplinary work, integrating the work of people who work in different areas. That is not something that is very well supported either by academic money, because the academic money goes into silos usually, and is audited by people who are themselves within very narrow domains, so it makes cross-disciplinary work more difficult. And outside of that, it's hard to do something that has a long horizon, because venture capital usually only gives money for short periods of time. So together with Ed Boyden at MIT, we're looking at this problem of architecting projects like that. How do we make it more interesting to people to invest in these problem-solution connections that are cross-disciplinary? And then I guess this is the end here. I've already told you all these things, so you can just read the summary. And thank you. That's my talk. I think at this point, I'm going to hand it over to the next person. And we're going to do this in a format where you all give your own little talk, and then we start asking each other some questions, talking around the table. And then eventually, maybe we can open up the floor to the public as well. Let's see if I can find this now. I'm trying to find how best to introduce the next speaker. I think the next speaker is going to be you, Mikael. Yes, I thought it was agreed you, and then Stephen last. Okay, so it's going to be Mikael. And as you already heard all of his credentials at Duke University. So I'll just let him say his piece now. Yeah.
Okay. So I will give you an introduction on brain-machine interfaces brain-machine interfaces that handle movements and handle sensations. I am from Duke University, and I'm also an editor at Frontiers in Neuroscience, where we run a research topic on augmentation of brain functions. So everybody who wants is welcome to contribute. It is very close to our topic today. But let me start with showing you the picture of a monkey brain. As you can see here, there are many areas which are interesting to the designers of brain-machine interfaces. There are representations of different body parts, different functions. So there are many places where we can put our electrodes and record neuronal activity. We work with two types of monkeys, rhesus monkeys and owl monkeys. They have slightly different brains, but the basic layout is the same. And it is also very similar to the layout of the human brain. So this is basically why we work in monkeys, because monkey's brain is similar to the human brain. So this slide illustrates the basic idea of a brain-machine interface. Here a monkey is playing a video game, and it starts with using a joystick to perform this task. And then we record brain activity, decode monkey's motor intentions, and then we make the brain control this video game directly. And also we send sensory feedback to the brain to mimic sensations. How is it possible to decode neuronal activity? Basically, we use the property of brain neurons that their firing rate represents behavioral parameters. For example, in this case, when the monkey moved the joystick to the right, the cell exhibited the highest level of activity. When it moved it to the left, the cell activity was low. Using this property, we can decode monkey's behavior from looking at this cell. Even by listening to this cell, you can tell what the monkey is doing. But of course, one neuron is not enough, and you need many neurons to be able to decode accurately. So we implant many electrodes in our monkeys, and this slide illustrates steps of an implantation surgery. Openings are made in the skull, and after that, we insert multi-electrode array in the brain. We fix it, and we have a monkey with a cap with a stick and connectors. So then all we have to do is just to connect our recording system to these connectors. This is one type of our multi-electrode array. We use different arrays. In some of them, most recent, we use electrodes that can be independently movable. And if we are lucky, This is what we record from our monkeys. Here we recorded around 390 neurons. Here each cell in this table represents an electrode, and each colored wave represents a neuron. And on every electrode we can have actually several neurons recorded. Now, after we recorded several hundred neurons, the question is what to do with this immense brain activity, how to decode it. And we use different decoding algorithms which work the following way. They look at the firing rates of neurons, then they use equations to process this activity. And at the output of this algorithm, we have a prediction of a behavioral parameter. For example, x and y coordinates of an arm movement. I will start with showing you a brain-machine interface that decodes locomotion. In Japan, there are trainers that train monkeys to walk bipedally. And this is a famous Japanese art. We are not Japanese, but we can also train monkeys to walk bipedally. And here is a monkey walking on a treadmill. It has markers on its joints, on the hip joint, on the knee joint, on the ankle. And we use those to track the kinematics of the monkey locomotion. And we also, as I showed you, record the activity of the monkey cortical neurons while it is walking on the treadmill. Then we train a decoder, and we can predict what the monkey is doing by just looking at the brain cells. Here, the blue leg represents the actual monkey movement, and the red one represents the decoded leg, which is decoded from cortical activity. And you can see that the red leg follows the blue one pretty well, which means our decoder works well. Well, if we can decode activity this way, and we can do this for slow movements, for fast movements, for variable speed movements, then what to do next? And what we did was to connect our monkey in Durham, North Carolina, to a humanoid robot in Japan. So the monkey was doing this in the United States, and the robot was in Japan. And the robot reproduced the monkey's movements. First, it was suspended over the treadmill, as you can see in this video. But then they improved their robot, and it could walk on the floor. So here, the monkey's brain activity controlled bipedal walking of a robot. Now we can also decode arm movements. In this experiment, actually conducted 10 years ago, but pretty famous, we trained the monkey to move a joystick on the screen, or to move a cursor on the screen, and the monkey was using a handle that it could squeeze to mimic grasp. So the monkey's task was to reach to a target and then squeeze the joystick handle. It was doing this, and we recorded brain activity, trained our decoding algorithm, and then we disconnected the joystick electrically from the system, and now the brain controls this. So you can see that the brain control is somewhat noisy compared to hand control, but the monkey can still manage it. So here we predicted three variables: position of the monkey arm and the gripping force. To improve our decoding, we started to use more advanced decoding algorithms. Kalman filter in this case looks pretty sophisticated, but it works better. And using these new decoding methods, we could now predict bimanual movements. Now the monkey was trained to move two virtual arms simultaneously. So one virtual hand reached to its target, and the other virtual hand reached to the other target. This is manual control, where the monkey is doing it with the 2 joysticks. But also, the monkey can do it simply by thinking. Here we record cortical signals, and the monkey directly controls 2 arms simultaneously. What else? We can decode from the brain while the monkey is not doing something, anything. In this task, the monkey was trained to press a button, then mentally time 3 seconds, and then release the button. And we could decode the monkey's processing of time. And with the recordings from hundreds of cortical neurons, we can do this very well. Well, we can decode. Now we wanted to send information back to the brain. So now we'll show you how we did this. We started experiments in owl monkeys. And the owl monkey had a very simple task. It had in front of it 2 doors, and behind one door there was food hidden. And we used implanted electrodes to apply microstimulation to the somatosensory cortex of this monkey. This stimulation told this monkey which way to go. We used different patterns of microstimulation. Some were pretty simple. It was either stimulate or no stimulation. But we also used different temporal patterns where the primary frequency of stimulation was the same, but the secondary was different, and the monkeys could do it. And also, we could use spatio-temporal patterns where we sent waves of stimulation across the monkey brain. And the monkeys also could do this. So we like this result, and we shifted to smart rhesus monkeys. And here we did an experiment which we called brain-machine-brain interface. We called it this way because we simultaneously decoded brain signals and delivered sensory information back to the brain. So the monkey had an array of 3 virtual targets. Visually, all the targets looked the same, but when the monkey touched these targets with the virtual hand, it received different patterns of stimulation in the brain. And for some patterns, the monkey was rewarded when it held this target. For some, the monkey was not rewarded. So this is how this experiment works. The monkey has 3 targets, they look the same, and it starts to scan it with this virtual hand. And when it places the hand on the target, it receives a microstimulation pattern in the brain, which feels like a texture. And the monkeys can do this by moving a joystick, or, like in this video, the monkey can do it by simply thinking about this through brain interface. Well, we did this interface. And the next step was to mimic the situation when the monkey scans virtual targets. Here the monkey has one virtual finger, and it scans targets which have different virtual texture represented by vertical gratings of different density. And the monkey has to rub this virtual finger against the targets and receive microstimulation pattern, and then identify the hidden virtual texture. It can do it manually with a joystick, or it can do it in a brain machine-brain interface mode, like in this video. Well, we did brain-machine interface, brain-machine-brain interface. What next? Next, we wanted to do experiments where we used several brains simultaneously. And this experiment we call brain plus brain interface, because we ask our monkeys work cooperatively, either 2 monkeys or even 3 monkeys. So we sum their brain activity, and using this summation, monkeys can perform advanced tasks. like in this case, there are 3 monkeys involved, and the final product of this brain plus brain interface is represented by this black dot, and they are actually hitting a 3-dimensional target using only 2-dimensional displays. What is next? The next development is brain-to-brain interface. So there is not only summation of activity of different brains, but there are also communications from one brain to another. So in this experiment performed in rats, one rat was doing a task. We recorded activity of this rat brain and sent the representation of this activity to the brain of the other rat. And the rat had to read out information from the other rat brain in sort of telepathic way, and then reproduce what the first rat was doing. Moreover, there was a feedback loop, because if the second rat performed correctly, then the first rat was given a reward, so they worked cooperatively. And recently, this experiment was reproduced in human settings. And at this slide, I will pass this microphone to Steve. Because,
okay.
Actually, no. Then he will talk about this probably later.
Yeah, actually, I think it makes sense if we do the introductions of each of their fields first, and then we have the discussion right after. So why don't we do it that way? Okay. So then I guess you're next. Yeah.
Is it working? Yes? Okay, well, I come about as completely from the other end of the spectrum, I would say, from Randal and Mikhail. Mine is a creative project, as Timur explained before, and to tell you a little bit about it, I won't go too much into the background, but I've been doing this for I initiated it about 10 years ago. That's there for I won't go into, as I said, I won't go into the background, but it came out of a chance meeting with the director of the Brain Sciences Institute at Swinburne University in Melbourne. And what evolved from that was me doing a small research project there and having a number of EEGs myself, and I simply couldn't resist that. So that gives a little bit of an idea of how my brain works. You know, when I saw the photo, I had to do something with that. But rather than dwell on how this came about, I'll tell you more about recent events. We seem to have skipped a few. For some reason, we're missing a little bit of the... it's not coming up. I have a technical collaborator who has designed a software. His name's Harry Sokol, and we've been working together for 10 years now. I create the visual input and come up with the ideas, and Harry actually writes the code for this program. So what it is at this stage is more of an authoring kit where we can use EEG data to create or control a series of effects. We've actually got it to a point now where we can, where it is actually real time. Sorry, I'm hearing a bit distracted from the sound out there. What we're doing is actually in real time. So we have some commercially available neuro headsets that were actually developed in Sydney, Australia, the Emotive headset that were originally developed for gaming purposes, but we've adapted this to actually do performance events and live installation works. This is the launch of the Global Mime project, where 3 performance artists were invited to respond to their idea of mind, I suppose. And each of them chose a very different way of actually tackling that. It's not necessarily reflective of their personal philosophies, but the artist down on the bottom left, Jill Orr, chose a very theatrical approach that was more like the sort of 19th century mentalists, and she had a couple of other people working with her, so it was a very interesting sort of approach that she did, and it was a very interesting collaboration with her. There were each of those artists, I worked directly with them, or we worked directly with them to interpret or to respond to the way they actually work as an artist, and each of them had a very different way of collaborating. In fact, they were quite extreme. One was the well-known performance artist Stella, come on the top left. He's quite well known for robotics and various other things, but his was very much about the whole augmented body and mind, you know. So I was perhaps unnecessary, you know, in the process, whereas the person on the right, who is a practicing Buddhist and does performance events, sort of endurance performance events, blindfolded, playing piano, was, I don't mind, Karen, I trust you, whatever you feel like doing, you know. So it was from one extreme to the other. Jill was very much a hands-on collaborative process between the two of us. So it was interesting just seeing how, you know, seeing how each of those artists work and how their minds ticked anyway. But I'll play you a little snippet of video just to give you a bit of a feel of that event. music with himself. We'd recorded his piano samples, and his brain was triggering them, and he was playing along as well. So. But it was a very interesting process how they all chose to work, you know. So, and the whole thing was an experiment. We had no rehearsal at all. It was just go in and do it. So I had no idea whether it was going to work or not, and that's pretty much the nature of what it is I do. Moving forward, this is. An installation work from the International Symposium of Electronic Art in Turkey in 2011. And this takes it up more to what it is I've been doing over the past couple of years. This particular work is being generated from the software live. However, the soundtrack was pre-recorded. The EEG data was also pre-recorded, my brainwaves. Need some volume. Snippet of what it is that work was about, but when I create a work elsewhere, I always like to take a little bit of the place that I've come from to another location and absorb a bit from there as well. So I don't know whether you could notice the soundtrack there. It had the influence of Australia with the didgeridoo and the Sufi ney, the wind instrument you could hear. So, and also again the patterns seem to begin to look a lot like Islamic tiles, but also that was very much like the desert in bloom after the rains in Australia. So how that imagery is actually created is I actually use my own artwork as well as my brainwaves to generate it. So they're quite often earlier works. I had a history as a painter and printmaker originally, and then I moved into video art and installation. So it's a way of actually using some of my old works in what I do now. But I'll move on to the next one, which is, it takes it a step further. The sound is actually being generated too here by the EEG data. I suppose this is sort of really stems from work I've been doing for a great many years, and a lot of that is creating spaces for people to, I suppose, experience some sort of altered state, you know, whether it's a meditative state or hypnagogic state. And I've been doing that for 20 years or more. Have I?
It's about the moment when yes, that's the moment to finish, I think.
Okay. Well, we'll leave it at that now. Yep.
Yeah.
I
Yeah.
wasn't watching you.
Thank you. Thank you very much. Okay, let's give you a chance here to hear. It's loud. Yeah.
Okay, I'm going to move around here because I can't see anybody from back here. My name is Stephen Dunne. And as Randal mentioned earlier, what we do is probably the most down-to-earth or most practical of what you've seen today. We call it applied neuroscience. Basically, it means we're physicists and engineers rather than neuroscientists. And what we do has to have an application in the real world. So to give you some background on why that is, we're a private R&D company, but our mission is to transform science into technologies that has a positive impact in the world. And impact for us means that there's a market. So we actually try to build things that we can sell. This is kind of our metric for success. Can we sell it to somebody and do something real with it? Okay, so what we mean by applied neuroscience is we don't do a lot of this. For us, the brain is more like this. It's nice and clean. We work only with data. Everything we do is data-driven. We very rarely get mixed up with what goes on the inside. And we don't pay a lot of attention to it. And I'll explain a little bit more about that later. So we work with the electrical brain, so basically signals from the brain. So what we do, we build technologies for use in the real world, as I said. There are two aspects. One is we read from the brain. We have a Bluetooth wireless cap, like this one you can see here, for reading from the brain. We have another one to write to the brain using non-invasive current stimulation. And if you put the two together, it opens up some interesting possibilities, which I'll come back to later. Another aspect, first of all, just to mention that the way we do this is we read using EEG, which is electrical. And as I mentioned, we stimulate using transcranial current stimulation, again, electrical. And the hard part here is understanding exactly what's happening when you do this. So we work a lot in the models of what's happening electrically in the brain. This allows us to not only understand what we're recording, but also to target the stimulation to try and affect specific functionality. So what I'm going to do now is just kind of give you an overview of what we're working on right now with some of our partners, basically all around the world. One nice project we have at the moment is for the Michael J. Fox Foundation on Parkinson's disease. So based on EEG alone, we're developing biomarkers for Parkinson's. We have a very nice result, a small group at the moment of 100 patients, but using EEG data recorded up to 8 years before they presented symptoms for Parkinson's, we can classify according to whether they will develop it or not. This would have like a profound impact on research in Parkinson's and treatment. We have applications in neurofeedback. The main one is attention deficit disorder, where you use brainwaves in real time to drive a very simple game. Kids with attention deficit disorder connected to the device controlling the game are essentially learning to control their brainwaves. To them, all they're doing is controlling something like the submarine you can see here, moving it up or down. But what they're actually doing is learning to control their brainwaves. And this entrainment improves their ability to control both their emotions and concentration. We have applications in user state. We had a very nice project called Beaming, where people were embodying an avatar similar to the idea Mikhail talked about earlier. And our work was to capture the emotional state of the user and transmit that along with the movement so that you could animate a robot on the other end, not only with the physical movements, but with the emotional state. So it's not a very fast control signal. It's a very slow emotional one, and we think BCI is very suited to this type of work. Another very similar application is stress measurement based on EEG. In brain-computer interface, we don't do a lot of work on actual control. Again, as I mentioned, it's more like emotional state. But this is a very nice application where there's an exoskeleton controlled by a BCI. So he's not controlling the movement of his arm. All he's controlling is the grasp, which is a very simplified problem. So he moves his own arm. He has an exoskeleton which closes and opens. And this single movement is controlled by the BCI. So we work on these types of applications. So all of these so far are read applications. What about the write, which is basically the stimulation? So in neuromodulation, our advantage at the moment is we have multi-site stimulation. We can stimulate the brain in up to 8 different locations. We do a lot of physical modeling of what's happening in the electric field, and basically the upshot is that we can control which part of the brain is being stimulated or which parts. This is a huge leap over what was the state of the art up until very recently, basically 2 electrodes, quite large, where you were stimulating or inhibiting basically all of the brain. Very hard to know what was happening, very hard to control which part of the brain you affected. We can now, for the first time, stimulate the brain using this multi-site, multi-electrode approach with modeling, and very focally control which areas are being modulated. So some applications of this, Adam Ghazali published in a very interesting paper in Nature last year, using a combination of video game design and stimulation for cognitive enhancement. So he was improving the plasticity of older patients using a combination of a video game and stimulation. And his results basically said that, I think he had a group of 65 to 75-year-olds that had the easiest way of explaining it is a level of plasticity closer to a 25-year-old's. So their ability to learn had gone back to the ability they had when they were 40 years younger. Another group we work with in Canada are using the device just for EEG as a consciousness scanner. So for locked-in patients or patients in coma, they're giving a quantitative measure of the level of consciousness. There's a lot of research on whether these people are conscious at all. So this is a quantitative measure of the level of consciousness based on brain activity. Another group in Barcelona at the Gutmann Clinic are using a combination of stimulation with visual stimulation. So they're stimulating the motor cortex while somebody is watching a video where the top half of the video that you can see here is their body. The bottom half is virtual. So this patient who's confined to a wheelchair is looking at a picture of herself or a video of herself walking while we stimulate the motor cortex. And this basically takes away the neuropathic pain that she's been suffering from for the past 20 years or so. Another clinic in Boston, the Spaulding Clinic, there was a case study. I say case study because when you're working with this type of injury, it's very difficult to replicate these results and get statistics. But in this one specific case, a 3-year-old who almost drowned, very severe brain damage, wasn't responding to occupational therapy, didn't recognize the voice of his parents, couldn't grasp objects, basically wasn't developing at all. They had given up. They didn't have any other therapy to try until we came along and suggested they use stimulation. And after a couple of months of stimulation, the kid is responding to his parents' voice, is able to grasp objects. We're not quite sure why. We're stimulating different parts of the brain. As I said, it's a case study, and until there's a lot more subjects of a very similar nature, it'll be difficult to say exactly what's happening. But in this case, it's working very well. In the Burke Rehabilitation Center in New York, they regularly use this type of stimulation in combination with physical rehab for post-stroke rehabilitation. And they're taking up to 3 months off the rehabilitation time on patients. So normally it would take about 9 months in physical rehabilitation to recover at least some functionality post-stroke. They're doing it in 6 months by stimulating the motor cortex while they're going through the standard physical rehabilitation. A very interesting one with the group at Oxford is cognitive enhancement. So now we're talking about healthy subjects, or possibly patients with dyscalculia, which is the numerical equivalent of dyslexia, people who have difficulties with numbers. they're stimulating the prefrontal cortex while they give them certain mathematical tasks. And they're seeing quite significant increases in performance after a single session of stimulation. So that's kind of an overview of what we're doing right now out in the real world. A lot of case studies, which means there haven't been clinical trials to demonstrate efficacy at the level of FDA. But because these are private clinics, they're free to try different techniques in a kind of case study scenario. So I would basically say there's a lot of very promising results out there with a combination of reading EEG and electrical stimulation, and a combination of the two in a kind of a closed loop scenario, similar to what Mikhail was talking about in the invasive scenario with monkeys earlier. As I mentioned at the start, our goal is to sell products, which in humans at the moment means non-invasive, which is why we're focusing purely on EEG and electrical current stimulation. Both can be done with a wearable Bluetooth cap. You can set these up in minutes. They're relatively very cheap for a medical system. Basically, you could do this at home on your own. So it's very, very easy to use this technology, and we think it has a lot of potential, not just in the medical market. But we're also a research company, so we have been thinking about what's going to happen next. I think cognitive enhancement is definitely going to be a big one, a combination of recording what's happening in the brain, measuring your cognitive performance on the one hand, using just EEG, so you have a quantitative measure of your performance. And this has direct applications in dementia, which would be our first target. So measuring on the one hand and stimulating for treatment or offset of dementia symptoms. Mild traumatic brain injury, such as in the case of the 3-year-old we saw earlier. But beyond that, we can think of healthy subjects, as in cognitive enhancement for mathematical ability. As a study aid, we know that a lot of students are taking prescription drugs for cognitive enhancement, particularly in the US. They might be very interested in a device like this, which has, as far as we know, no secondary effects whatsoever. It's been used for 35 years without any reported serious secondary effect. And sports. There's a lot of interest in the area of sports as an aid to improving concentration, getting in the zone, this type of thing. But beyond that, could you cheat and put yourself in the zone using stimulation? So the term neurodoping has already come up, and there's been a lot of discussion around this, and should it be regulated before it becomes widespread. So we think sports performance in general is a big area of interest for us as a company. We're already talking to different groups about this. This is the Human Performance Lab in London, funded by GlaxoSmithKline, and they already have a cognitive wing in sports performance, and they work with some of the top athletes in the world. Right now, they're not using EEG or TDCS, but they are interested in cognitive performance. And then the last one I want to mention is what Mikhail mentioned very briefly earlier on, is brain-to-brain communication, which was basically a replication of what he did in monkeys and humans. We published this last August. Mikhail knows it very well because he was the editor in +1, which was where we published it. And the idea here was to see if we could transmit information from one brain to another, bypassing all of the senses. So the transmitter was imagining moving their hands or moving their feet. And that was our 0 and 1 for the information we were going to transfer. So they weren't using any of their senses to transmit information. They were just imagining using their hands or using their feet. The receiver, which the transmitter happened to be in India at the time, so this was done from India to France. The receiver was in France, and there we wanted to use non-invasive stimulation. In this case, we use magnetic stimulation rather than our own cap, which is current stimulation, because it's more powerful. But in order to bypass all of the physical senses, and to make sure that they couldn't feel the actual stimulation in any way, we stimulated the visual cortex, and we produced what are called phosphenes, which is like a flash of light, but it doesn't involve the eye. So you're stimulating the visual cortex at the back of the brain directly. So we had a transmitter and a receiver that weren't using any of their physical senses to transmit very basic information, ones and zeros, and we coded a message in the ones and zeros, which happened to be hola in Spanish. So it was a very, very simple experiment and a very basic proof of concept, but it was the first time in humans that we had brain-to-brain communication, and the media loved it, and they jumped on it and called it telepathy, which we never mentioned in the publication. But after a month of having media call you and say, is this telepathy, we kind of finally gave in and said, okay, if you want, this is telepathy, or digital telepathy, to be more precise. But it's just a proof of concept, and it's very clumsy as a way of communicating, but it really just kind of opens the door. And if you think about what we've already achieved in a very few years in terms of EEG and non-invasive stimulation and how quickly this is improving, you can imagine that you could do something far more subtle and far more interesting with a wearable Bluetooth device in a matter of years, which could open up a whole area of research, which for us at the moment is in the proposal phase. So we've submitted a few proposals that are linked to things like group dynamics, when you could have a lot of people interacting like this. In thinking about things like synchrony, how well people play musical instruments when they can see each other together. What if they can't see each other, but their brains are synchronized? Things like this. So I think it's going to be a very, very interesting area. And we're submitting a lot of research proposals on this topic right now. And if we're lucky, maybe starting from next January, we'll be working directly on these topics. And this is the kind of experimental setup just to show you how that actually works. So on the left, very simple BCI, 0 and 1 transmitted to a guy in France using this large robot to control a quite simple stimulation device. The robot was needed for the precise location of the stimulation. And that's all. Thank you very much.
Time, so I guess it's best if maybe you say a few words about where NeuroWeb comes from, and that will be how we finish the session.
So I will also speak in English. I'm the only person who is not a neurophysiologist here, or directly working with neurophysiology, but also probably Karen, you, coming from a little bit different walk of life. But my background is a combination of innovation studies and interest in education. And I work with the theme called road mapping and foresights for the last 6 years actively. And my interest was the future of education. So I was actually working with Russian educators, trying to understand where the situation in Russian, well, generally in education is going, what will be the long-term perspectives of education. We realized that in terms of technological transformations, there will be a major breakthrough if something becomes possible to actually directly work with neural tissue. So in the extreme manifestation is something that you can see in the movie Matrix, when there is the main hero, Neo, actually plugs in, I don't know, an implant, and it downloads all the lessons in a flash of a second. It's a very artistic representation, and probably it will not be possible at all, knowing to how brain operates. Maybe it will be possible if Randal's project succeeds and the brain is replaced gradually by some artificial components. But at the moment, how we know the skills is formed. You remember this part of the movie when he downloads the knowledge of Kung Fu in his brain and immediately is able to fight. And yeah, but if it's something like that happens, it will completely replace the pedagogy with something new. So we wanted to look, we realized it will be the end to all of education, and it was something new will come up. So, but as we looked into this area more deeply, starting to work with Russian neurophysiologists and psychologists, we realized that it opens up the whole new world of possibilities beyond education. So we can think of applications in management, in design of products, in arts, in video games and entertainment, in control of robotics in industries, in military applications, and so on. There's a whole host of new applications possible if we are able to read brains, if we're able to write brains, and in precisely, because as you have seen from discussion today, the frontier of neuroscience is mostly with locomotive ability, which is probably the most crude out of all. When we're talking about ability to comprehend languages, to think, to read specific images that emerge in the brain. Well, this is also partially possible. For instance, reading from the optical cord that connects our eye to the brain. But more sophisticated type of reading, the higher neural functions, as I understand, we're really quite far from that. But again, going into that direction, this is where a lot of investment already happens. And this is where the science is going, where the technology is going. I think a lot that Stephen does in practical terms is actually also moving in that direction. So the question, of course, emerges: what are the scenarios that are desirable? What are scenarios that are not desirable? Because if you think about reading minds, for instance, directly reading from neural tissue, this means that if it is possible to decipher information from the neural tissue, it can be put to really bad ends as well, like controlling our minds by the governments or major corporations, which is totally undesirable. So this is, this is very, very far from where we are now, but we obviously going into direction where it becomes more and more possible. So the question is, how do we, going long term, can create a roadmap that leads us into sort of desirable futures and a little bit away from the dystopian futures. So that was a sort of long-term inspiration for the neural web. And again, we cannot speak only about this only philosophically, only in terms of like very, very far long-term possibilities, but we have to also think about the feasible things, like applications that are possible in next 3 to 5 years or next decade. And also the important aspect is to integrate different scientific groups to collaborate on these positive scenarios. So the creation of communication through the internet, as you have seen from the last description, it already becomes feasible with a very simple type of information from one brain to another, but it is already a proof of concept that exists at this moment. So there have been first experiments that prove it is possible. So this means that it really opens the world for connecting brains to brains and transferring more and more complex information from brain to brain. So this calls for some kind of coordinated effort to actually build up a sort of a roadmap. And I think the way that, for instance, Randal approaches in his work is an example of how this kind of work can be done without pushing the agenda on researchers, because it's not possible to push it on them. They're all independent. They work from different sources. They work towards different goals. But it's also, if our researchers understand that there is a possibility of creating a big and connected type of project, then there can be a shared interest. So this is the inspiration for NeuroWeb roadmap that we're trying to create as of now. And actually, we believe that it opens also a possibility to actually support the development of large new industry. Some parts of this industry will emerge because there is big demand from medical industry, for medical sector, but other parts are not as guaranteed, so to say. So the question is, how do we coordinate efforts of researchers and practitioners in this area? And I, by the way, believe that science artists like Karen will be very important part of this movement, because they serve as pioneers in applying these tools to the real world, maybe in very radical ways sometimes, but it's very good way to explore those boundaries of applications, see where we can go together. So this is actually why this group is here. we would like to continue this work further. We will have a workshop tomorrow, which actually is contributed to dedicated to this collective work. We will try to see how we can connect our work and works of other groups that we know of. So, and hopefully our intention that Russia can become part of this movement. There are several strong groups in Russia. For instance, the laboratory of Alexander Kaplan at Moscow State University that can be part of the, well, they're partially already part of this international dialogue. And going forward, it is important to do roadmap, to start coordinated research, to do prototypes, to do applications, and do it in a more coordinated manner. So this is what we would like to do going forward. Thank you.
Thanks. We've run out of time, so I'd like to thank all of the speakers, and I guess this is how we're closing the session. Thank you all very much for being here and listening.